Hello, my name is Kathy Hernandez, and I will be presenting on the TP53 gene. This is one of the genes that are listed on the figure. This is the tumor suppressor protein, P53. So TP53 gene is also known as the guardian of the genome. TP53 encodes for the TP53 protein or P53 protein. This is a transcription factor that is critical for tumor suppressing in cells. Overall, P53 helps maintain genomic stability and prevents the formation of tumors. It is often referred to as the guardian of the genome due to its ability to control various cellular responses to stress, including DNA damage, oncogene activation, hypoxia, oxidative stress, and nucleotide depletion. So, when functioning properly, P53 can trigger a variety of protective cellular responses, such as DNA repair, cell cycle arrest, apoptosis, and senescence. So, apoptosis is also known as programmed cell death, and senescence is permanent cell cycle arrest. So P53 plays a crucial role in cell cycle regulation. We can also see that in the figure to the right, P53 and cell cycle arrest. So P53 mediates cell cycle arrest in response to stress signals, which allows cells time to repair DNA damage or prevent the propagation of damaged DNA. So at the G1S checkpoint, P53 arrests the cell cycle in response to DNA damage, and this delay allows the cell to repair the damaged DNA before proceeding to the S phase, which is also the DNA synthesis phase. So P53 achieves this by activating the expression of P21. This in turn inhibits the activity of CDKs, which is necessary for progression through the G1 checkpoint. So this arrest prevents the cell from entering the S phase with damaged DNA and then protects the genome from further mutations. Then at the G2 mitosis checkpoint, if DNA damage is not repaired in the G1 phase, P53 can also induce a G2M cell cycle arrest by activating other downstream targets. So this provides an additional checkpoint before the cell divides, ensuring that the DNA is intact before mitosis. If the DNA damage is severe or if it's irreparable, the cell is unable to repair it. P53 can drive the cell into senescence, which is a state of irreversible cell cycle arrest, or initiate apoptosis, which is programmed cell death, to prevent the proliferation of damaged cells that could lead to tumor formation. In the article, P53 promotes cell survival due to reversibility of its cell cycle checkpoints. Researchers investigated P53's role in cell cycle regulation and checkpoint reversibility. So this part of the study that is depicted is figure 1. It's also titled Loss of P53 Enhances Tumor Cell Death in Response to Continuous Chemotherapeutic Treatment. So in this part of the study, researchers investigated the role of P53 in regulating the cell cycle in response to DNA damage, and in order to understand how the loss of P53 affects the cell cycle and cell survival using chemotherapeutic treatment. So the experimental design created by the researchers investigated P53's role in cell cycle arrest. The U20S cells were transfected with control shRNA and P53 shRNA vectors. So stable expression of control shRNA and P53 shRNA clones were observed, and each underwent a treatment with doxorubicin and camptothecin. Then flow cytometry was used to analyze the cell cycle progression. So this setup allowed the comparison between P53 proficient cells, which are the control shRNA, and P53 deficient cells, which are the P53 shRNA. This tested whether the loss of P53 impairs the cell cycle arrest and induces cell death following DNA damage. Researchers used chemotherapeutic agents such as doxorubicin and camptothecin in order to induce DNA damage, and this would trigger a cell cycle checkpoint response. So in figure A1, we're looking at immunoblotting of P53 and P21 expression. So immunoblotting was used to detect the expression of these two because P53 and P21 are accompanied genes. So after doxorubicin treatment, control shRNA cells showed P53 induction and an accompanied increase in P21 levels. So these results are consistent with the expected role of P53 in causing cell cycle arrest by inducing P21. This verifies whether P53 activation and the subsequent induction of P21 are required for maintaining cell cycle arrest after DNA damage. So the result for part 1A is that P53 shRNA cells do not no induction after treatment. P53 response to lighting. So in figure 1B, we're looking at flow cytometry analysis of cell cycle distribution after rubicin treatment. So we're looking at 1 day, 2 day, 3 day, and 4 day, and 6 day. And then the flow cytometry was used to analyze DNA content and measure the distribution of cells across different phases of the cell cycle, which are G1, S, G2, and M. So the DNA content of the cells was assessed at different time points after treatment to determine if the cells were arrested at G1 or G2 phase. So the control shRNA cells exhibited a normal DNA content distribution with the 2 day and 4 day arrest upon doxorubicin treatment, suggesting a sustained cell cycle arrest. And then P53 shRNA cells showed a transient 4 day arrest, followed by a hypodiploid peak later in the treatment period, which indicated cell death. This demonstrated that P53 deficient cells failed to maintain the arrest and led to cell death. So some conclusions from A, B, and C were that P53 is essential for maintaining G1 and G2 arrest after DNA damage for preventing cells from progressing through the cell cycle with damaged DNA. And in the absence of P53, whether through stable or transient silencing, cells fail to maintain their arrest and undergo cell death. In this article, we're looking at the mutant TP53G245C and R273H, which promote cellular malignancy in esophageal squamous cell carcinoma. So we're looking at the mutant TP53G245C and R273H point mutations, which lead to the disruption of cell cycle and uncontrolled cell division. So in figure 2A and 2B from this article, MTS assays were used to test the impact of mutant TP53 on cell proliferation. The different characteristics of various TP53 transiently transfected cell lines were analyzed. So the different names of the cell lines are on part A, the KYSE150, COLO680, H1299, and HCT116P53. And the mutant TP53 led to a significantly greater promotion of cell growth than the wild TP53 transfectants. Compared with the control cells, wild type T53 suppressed cell proliferation, while the TP53 mutated G245C and R273H cells strengthened cell proliferation abilities. So the mutant TP53G245C and R273H point mutations led to the disruption of the cell cycle and uncontrolled cell division. So MTS and colony formation assays showed that mutant TP53, G245C, and R273H increased cell vitality and proliferation.